Hello 12th class student. In this video we will study about the test cross. In the previous videos we discuss all the Mendelian laws and exceptions of the law of Mendel. Now here we will start with the test cross and the back cross. Firstly we will discuss about what is the test cross. Student, this is a method which is used by the Mendel to determine the genotype of an organism. At that time Mendel do not know about the genetic material. He don't know the DNA, so he gives the term factors and alleles. There is no availability to check the genotype of an organism in the laboratory, so he identify the genotype of an organism by using this test cross. In this cross, the organism with dominant phenotype, but that organism genotype is unknown, is crossed with the recessive individuals. Let understand this by using an monohybrid cross. There are two cases can be there, case 1 and the case 2. In case 1, and here we will study the case 2. Let's study case 1 first. In the both cases, genotype is unknown to us. We don't know the genotype, but phenotype is known. Here we will take the parents. You know, in this test cross, one dominant phenotype is crossed with the recessive individual. Let me take the trait for the plant height. Here we take the dwarf, and here we take the tall. If in case this tall is homozygous, let's take this tall. If this tall phenotype is the homozygous, then let's perform the cross. Here we have to write the gametes. Both will get separate due to the law of segregation. Now F1 generation, capital T small t, capital T small t, capital T small t, capital T small t. 100% you will get the tall. All these are the tall. Due to the law of dominance, 100% F1 generation is tall. In case of if your organism is identifying organism is homozygous. So अगर आप चाहते हो, अगर आपको ये इस organism का आपको जीनोटाइप पता करना है, इस organism का आपको जीनोटाइप पता करना है, अगर ये प्योर डोमिनेंट है, अगर ये प्योर डोमिनेंट है तो 100% आपको टोल मिलेगी. Let's study case second also. If this organism is the heterozygous, let's study parents. One is tall, we have to cross this with the dwarf. Dwarfness is the recessive trait. If this is heterozygous, then gametes, capital T small t, here both of factor will get separate. F1 generation, this T will get crossed with this capital T small t. It will cross with this capital T small t. Here capital T small t and small t. What you see here, 50% of F1 offspring are the tall and 50% are the dwarf. It shows that if your identifying organism is the heterozygous, in that situation you get the 50% tall and 50% dwarf. So you can identify the genotype by using the test cross methods. Okay, so अगर आपने जो organism आपको जिस organism का genotype आपने find out करना है, अगर after the test cross it shows the 100% dominant trait, then you can say genotype is the homozygous, and if show the 50% dominant and 50% recessive as like this, then you can say that this is the recessive. Sorry, this is the heterozygous dominant. Now we will study about the back cross. We generally use the back cross in many individuals in the horticulture and the animals breeding program also. In back cross what we do is we do breeding or cross बिटवीन द F1 प्रोजेनी, जो F1 प्रोजेनी होता है एंड वन ऑफ इट्स पेरेंट, कि F1 प्रोजेनी और किसी भी एक पेरेंट के बीच में जो क्रॉस करवाना होता है, दैट इज नोन एज द बैक क्रॉस. हेयर इफ वी परफॉर्म ए क्रॉस बिटवीन द F1 प्रोजेनी, अगर ये एक F1 प्रोजेनी और इसके एक पेरेंट्स, किसी भी एक पेरेंट्स के साथ हम क्रॉस करवा रहे हैं, F1 प्रोजेनी का क्रॉस F1 विद द पेरेंट्स, दैट इज नोन एज द back cross. We perform this cross to get the more parental type genotype. अगर आप एक F1 का किसी भी पेरेंट के साथ क्रॉस करवाते हो तो जो नेक्स्ट जनरेशन होगी उसमें मोस्टली करेक्टरिस्टिक्स कैसे होंगे उस पेरेंट्स की टाइप के. ओके, इफ यू हैव एनी डाउट यू कैन आस्क via the e-feedback or in the comment section. Till that, revise all the crosses and learn these. In the upcoming video we will study about the pleiotropic inheritance and the blood group in human beings.